Times are changing. Yesterday's science fiction is today's reality, and the things we used to care about don't seem to matter as much anymore. So will nothing ever be the same? Well, not everything, because some things never change, like wanting the finest in care. We all want the most well-trained people on our side, to be seen by leading experts, passionate about what they do, who search for and find solutions, who are invested in their work. We at IVI Global Education believe in lifelong learning. That's why every year we help physicians, nurses, lab technicians, researchers, and patient information staff keep pace with the field, offering master's degrees in partnership with prestigious universities, as well as over 60 online courses available through our online campus, so professionals can stay abreast of the latest scientific developments. Think about it. Reproductive medicine is a constantly changing field, and one that is filled with opportunities. If you want to discover how far you can go, just visit our website and select which area of specialization you'd like to start with. EV Global Education, innovative minds, successful careers.
Good afternoon, everybody from Valencia, Spain. My name is Nicolás Garrido. I am the director of EV Foundation and also the co-director of this master's degree on the Biotechnology of Human-Assisted Reproduction and Embryology. It's my great pleasure welcoming you all on behalf of IVIRMA to this open door session where we are going to show the program of this postgraduate course, and the session will start with my lecture, the introduction to our program, and will be followed by Dr. Nuria Pellicer, who is MD, PhD specialist in reproductive medicine at EV Valencia, that will talk about the clinical practice in assisted reproduction from the doctor's perspective, and will be followed by Dr. Marcos Meseguer, who is PhD and scientific supervisor at EV Valencia in Spain, that will talk about the day-to-day embryology laboratory work. We will end the session with a short time dedicated to the questions that the audience may bring concerning the master's degree we are here presenting. And we are going to start with the presentation of the program. Before, I would like to start with a general overview, introduction to our company. Ours is a company dedicated to provide with assisted reproduction treatments that was started in Spain about 30 years ago, and now it's present in a number of countries. We have about 70 medical clinical offices, about 30 laboratories that are based mainly in Spain, but in some other European countries such as Italy, United Kingdom, Portugal, also in Central and South America, and mainly in the United States, where there are about 19 reproductive centers. So the session for today is regarding the master's degree on biotechnology of human-assisted reproduction and embryology. And I also entitled this session as the importance of continuing education and regulated education to offer the best service to our patients. From the very beginning, we always kept in mind the answers to this question. What is our responsibility to our patients? We always would like to be able to offer them the best diagnostic and treatment alternatives. So what we want from our professionals in assisted reproduction is mainly having a solid theoretical knowledge and also skills based to ensure performance, to be able to offer them the best diagnostic and treatment alternatives, because we feel we have the moral and ethical obligation to offer them the best, and for that it is necessary to be updated. There are many difficulties related with being updated, and the main difficulties in this sense is the speed in the evolution of knowledge. There's a lot of information coming out every day, and that it's often difficult learning from one's own experience, from conducting one's own research, in the sense that the expertise is not always available and the experience is only linked to high numbers of assisted reproduction treatments. So there are many centers which the low numbers of treatments that they are providing do not permit gaining experience and improving in this sense, or even conducting research. In some other cases, there are difficulties accessing formal training. There are many countries where there are no regulated studies covering this topic. What about choosing formal or informal training? Learning by imitations will prevent development. So it's of utmost importance trying to learn from a structured and a formal program. IVIRMA's experience in the international training of professionals in the field of assisted reproduction can be easily checked in the website from ivglobaleducation.com. There you can find not only this course, but all the courses and masters that IVIRMA is providing worldwide. And this is translated into the facts that we have been training and teaching thousands of trainees for almost 3 decades. And this only means we have a huge experience on this topic. Concerning the master program that we are now presenting is the evolution from previous postgraduate courses. We started about 27 years ago with the first master's degree that was mainly focused for gynecologists. Then, as the ratio between gynecologists and embryologists was changed, we split this program into 2 different and more focused programs, one for medical doctors and the other one for lab specialists. This was converted into an on-site 2 years master's degree. And afterwards, this resulted in the request from many people to develop our own online research and online teaching program in order to provide with this information and these possibilities to many people, not only in Spain, but all over the world, that were not able to attend any similar program like this. Now, in its 10th edition, this is the slide showing the main screen of our master's degree from our education website. As you can see, it's fully online and provided by our specialists, mainly from IVI Valencia and IVI Foundation. You can search and find all the information available on our website, but mainly the course objectives are first trying to cover the needs that many countries have on well-prepared specialists to work in the assisted reproduction fields. So in the last decades, there has been a growing demand of experts in this field, and related with this high demand of experts, there's a high need to train and prepare them properly. So through the e-learning platforms and these methods, the students are able to learn from their places with the maximum time efficiency and flexibility, besides having the opportunity to share their knowledge with some other students and experts. Through the virtual classroom, the students will have complete access to all materials and resources related to achieve and improve their knowledge, and will have close communication and personal monitoring from the experts from IVIRMA. You can have access to classroom sessions from anywhere through our webinar systems, and you can see it as many times as you might need. I think the justification and the objectives of the master at this point are clear. The program consists in 16 different modules concerning and including all the topics, not only from the medical procedures and the doctor's perspective, but mainly from the embryology laboratory and andrology laboratory perspectives, together with a huge background on research and focus on research, but also discussing the ethical and the legal aspects of human-assisted reproduction techniques. Among our professors, we can have such high profiles as Dr. Antonio Pellicer, which is one of the founding partners at IVIRMA Global. He is our CEO currently, and also is a full professor in the University of Valencia, the School of Medicine at the University of Valencia. He's a well-known author who has published about 1,000 papers in peer-reviewed journals, and is frequently present in all the main congresses in our field. Dr. Nuria Pellicer from the Human Reproduction and Endoscopic Reproductive Surgery Unit at IVI Valencia, and also working on the research at the IVI Foundation, will be one of the coordinators, mainly on the medical doctors modules. Professor José Remoí, which is another of the founding partners of IVIRMA Global and also full professor at the University of Valencia in Spain. Dr. Elena Labarta from IVI Valencia. Dr. María José Escribá, which is a senior embryologist in the IVF laboratory in IVI Valencia, also a well-known researcher. Dr. Francisco Domínguez, which is lab manager and leader of the research group on endometrial receptivity and embryonic competence at IVI Foundation. Irene Cervelló, the other lab manager at IVI Foundation and leader of the group of endometrial stem cells and bioengineering research. Dr. María José de los Santos, who is the director of the Clinical Embryology Laboratory at IVI Valencia. Dr. Amparo Mercader, one of the senior embryologists at IVIRMA Valencia. Dr. Marcos Meseguer, who will be with us later on in this session, which is the scientific supervisor and senior embryologist at the IVI Valencia IVF unit and one of the most known researchers from IVIRMA. Dr. Patricia Diaz-Jimeno, research group leader in genomics and systems and reproductive medicine from IVI Foundation, and myself as a part of the bunch of professors in our master's degree. Concerning the contents and the information provided at this master's degree, all the students will have access to the written tests in order to get downloaded and study the different modules, but also we'll have online access to recorded classes where the professors, through PowerPoint presentations that are spoken, will go through the different topics and the different lectures covered in each one of the modules. These are some examples of how the slides look like. So within the virtual classroom from the University of Valencia, we will keep all the texts, all the recorded classes, all the self-evaluation questions for each topic, all the test responses templates, all the commented answers to the exams, and all the documents of interest for the students. In the next few slides, I will show you how the virtual website from the University of Valencia look like, where all the students will accede in order to look for the classes and download all the materials to study, and also to conduct the exams and the tests. So the only required material is an active email address, looking periodically to this email in order to look and detect from the communications from the master's degree and from the professors, and mainly using the tools of the University of Valencia for e-learning. Considering the evaluation of theoretical knowledge, the questions after each of the modules, it's weight about 30%. There will be one final exam, including the full contents of the master's degree, which is worth 40% of the total mark. And then there will be a final master degree thesis, which will have a 30% value of the overall marks achieved in the master's degree. In this sense, for conducting this final master degree thesis, all the students will have the detailed information about how to proceed on this, but will be mainly focused into a bibliographical review of a specific topic. And I would invite you to check on the IVIRMA Innovation website all the publications, all the papers that our personnel has been publishing through the years. You can search by name, title, investigation area, and year. And I will recommend you to go through ivirma.com/innovation, where you will find all the information considering the research and results and scientific productivity that is produced month by month by IVIRMA. So, reaching the end of this presentation, why do we feel this postgraduate course and not another one? Why you should choose this and not another? First, I think because all professors, or a huge number of professors, can be considered as world opinion leaders. They are professionals that are well reputed, well known worldwide, publishing a lot of papers and being present at the main congresses in our area. But in relation with this, because there's fully updated information that directly results from the generation of knowledge that we achieve through the research and the technology surveillance that we are conducting. So you can expect from these masters the last concepts, the ultimate information, the most recent procedures and technologies available out there. For who and why this is prepared? Well, there are typically two different kind of profiles that accede to this master and these studies. First, assisted reproduction professionals, so people that is already working in some clinics that lack any formal training, theoretical training on the specialty that they are working. This is not real. This is quite frequent. And in some cases, they feel they missed the fundamentals, the basis of the knowledge in order to improve the way they do the work. Typically, people tend to be hired. Some experienced professional tend to explain how the process are, and they just repeat mechanically without knowing the physiology and the basis of all these processes. This avoids further progress and development of the work. And also in some countries, because there's a request, we are finding that some countries are now requesting people who even have some or a wide experience on assisted reproduction laboratories to update the academic background by some studies showing that they are prepared to do the tasks. And also for graduates that are interested in their professional development in assisted reproduction. So there are many cases that can be benefited from these studies. So the way to contact us is through the innovation website, through the education website, and directly you can just submit an email or you can ask some question in these sessions that we will gladly answer and contribute to solve. Before I leave the floor to my colleagues, I would like to invite you to the 9th International IVIRMA Congress that will be held virtually in November from 15th to 19th, that I think I'm pretty sure that will be of interest of all of you. So I will see you again a little bit later in the Q&A session, but I would like to leave the floor now to my colleague, Dr. Marcos Meseguer, in order to explain the day-to-day in the embryology laboratory.
Well, thank you very much for the introduction. And again, thank you for the opportunity of sharing with you the experience looking into the lab. And what we have done, like embryology, is like a virtual day. So I have prepared for you some videos showing how is the lab in the day by day. So I have done several videos to show you how is our organization and where is the place that we normally used to work. So first of all, I just take my camera and we go through the lab and through the outdoors of the lab, where the two main theaters are, or the main entrance of the lab is there. You see IVF, like the name of the lab in Spanish. So we are actually a big laboratory, which is also including outside of the lab one important desktop or place for working, in which we have the laboratory director, which is always close to the day-by-day work. And together with the main lab, we have a very big main entrance with a picture of a baby, which is always very encouraging for our patients when they come to the clinic to do an IVF cycle. And you see there how the elevators are placed just for the entrance of the patients from the rooms. In the lab, we have also additional laboratories like this one that we use for the PGTA. We do here the tubing and the fixation of some cells if we do fish analysis, which is very unusual. And we have two additional places, one for preparing the shipments of samples of embryos and oocytes to another clinics. So we have a room only for prepare this. And together with that, we have another room in which we prepare all the sperm samples, which is in front. So let's say that even that we have another place for sperm preparation, in the lab, we need a special place to prepare all the sperm samples that we need for our IVF cycles, which is relatively close to the lab. In this lab, we have flow cabins and we have also centrifuges. Because we need the theaters surrounding the lab, we have two main theaters. In those theaters, we used to do in the past the embryo transfers. Actually, we do not do it. We have a window to see the lab from outside. It's always interesting if some visits are coming and they want to see the lab at least from outside. And the theaters with respirators and with all the instrumentation necessary is just beside the lab. And it's very important because we're going to provide the embryos and we're going to provide the oocytes. So you see the main entrance and you see like we have like a front door or a window that we can open or close depending on what we are doing and depending also if we have visits outside or patients that maybe will not like to see what we are doing in the lab. We are inside the lab. This is in the early afternoon, so you see that actually we were cleaning. We have like an island with all the time-lapse instruments. We have there 3 embryoscopes, and in the other side we have 3 Jerry's time-lapse incubators. These incubators are making recording of the embryo development during 6 days. And we have also the box incubators to have the dishes ready for embryo culture. If we go to one side of the lab, we see these cabins called isolates. In these cabins, we do oocyte retrieval, and they keep the oocytes in perfect conditions of temperature and CO2. And it's very close related with the theater in which we get the oocytes or even the sperm pickup if we do testicular biopsy, or the oocytes are collected there. And immediately from that place, from that window, we get it inside of these isolates. If we continue to this side, we see this another big incubators to keep the oocytes during oocyte retrieval. And we see the ICSI workstations together with the isolates for working. So beside of each ICSI workstation, we have one isolate. This is another isolate for another oocyte pickup room because we can do 2 oocytes pickups at the same time in 2 different theaters with this direct window again inside to the theater. We keep going and we see you can see how it's possible to have this huge amount of workstations, of ICSI workstations or isolates which allow us to do a lot of things at the same time. At the end of the lab, we have the place for vitrification. Those are different cabins. Those cabins are fully open and they are with cold surface. So we keep this part of the lab, this corner of the lab, only for vitrification. We have our own laptops, small laptops, to work with the data with the EMR of the patients. It's a paperless lab. And you see at the end, well, this is an elevator with an isolate cabin that we take it for moving the embryos from the laboratory directly to the room of the patient. In that corner, we have another time-lapse system, is the EmbryoScope Plus, a more advanced version of the EmbryoScope. And beside them, the benchtop incubators that are in stainless steel places, steel tables. In each one of the benchtop incubators, these are Aztec incubators, we have the capacity of 6 chambers or 6 patients, independent patients. We have another EmbryoScope there. So we have in total 9 time-lapse incubators. We have 5 EmbryoScopes, 2 EmbryoScope Plus, and 3 carries. This is the other place for Aztec incubators. Again, you can see the huge amount of incubators that we have. Even we have an ESCO incubator. We keep those normally for embryo warming, and always close to them the box incubators to prepare all the embryo culture dishes, and also to prepare, you see there, these towers of dishes that we prepare and we keep in the box incubators to stabilize, to make the culture of the embryos. So we're moving to the end, to the last corner of the lab, in which we have also box incubators, we have an isolate, and we have also two microscopes to do PGTA with laser. Well, together with the lab, also we have an important situation, which is the possibility of do embryo evaluation by image analysis. And we have actually one of the biggest labs in the world with this technology. So I take the opportunity to bring to you some videos about the embryo evaluation and selection using this technology. So we have several platforms for embryo analysis, fully automatic or semi-automatic. One of the platforms that we have is this one that is here, which is the platform of the Jerry. The Jerry has 2 versions, the Assess 1.3 and Assess 2.0. And in these videos, you can see how we are following up the embryo development, and all the stages of embryo development are annotated and follow up with this technology. So we have the Assess 1.3 and we have the Assess 2.0. The difference between these 2 systems, the 1.3 and 2.0, is that one is partially automatic and the other is semi-automatic. Well, this is a software which is in the Gary Connect and Assess. And let's move to the other system, which is the EVA system. It's included also in the Gary Connect and Assess. And the difference with the other system is that including in the evaluation of the embryo, we have a fully automation of the classification of the embryos or embryo score. And the embryos are scored from 1 to 3, depending on some features that are evaluated fully automatic. So this is the first totally automatic system which is available for embryo analysis, and that is included in this Jerry incubator in the version of Connect and Assess. So you have seen the 2 options that we have available for Jerry software for doing embryo analysis, totally automatic or semi-automatic. We have also the EmbryoScope and the EmbryoScope software, which is called Viewer, Embryo Viewer. The EmbryoScope has a semi-automatic system for embryo evaluation. All the events of embryo development are identified by the system and are suggested to the embryologist to be confirmed. So we need to go step by step through every event of embryo development. We make the annotation, and then this is recorded for the final classification. So it's partially automatic because we don't do the annotations. We do the annotations manually, but we have a suggestion of the timings of the events by the system. so it's not completely automatic. But even it's telling us which is the morphology of the inner cell mass and trophoctoderm, and it's making us a classification of the embryos in 4 categories, or in 10 categories, sorry, from 1 to 10, which is a final evaluation of the quality of the embryo. So we don't need to take any other decision that select the embryo with the highest score. This is the version 3 of one software called KidScore. Well, we have the last system that we have in the lab is the IDA score. This is the first system that is using AI for embryo selection. So in this case, we don't need to do any kind of annotation. The software is doing a follow-up of the blastocyst development, and is giving us a score again from 1 to 9.9, which is potentially helping us to select the best embryo. So immediately, just in 1 minute, we don't need to do anything else. We have a score that we can apply for selection. So it's the first time that AI is applied clinically in a software which is available commercially and that we are testing in IVI in Valencia. So you have seen several of the platforms that we are actually using for embryo evaluation and selection. And that's more or less was my intention with this presentation. And also, well, Nicolas was showing you some of the embryologists that are actually working in the lab, and most of them will teach you in the master. And they are researchers also doing active research and publications in the field of embryology. And I'm looking forward to see all of you in the following sessions of the master.
Hello, everyone. Good afternoon. My name is Nuria Pellicer. I'm a gynecologist, a specialist in assisted reproduction. And today I'm going to talk about our day-to-day patient care in assisted reproduction. Also, I'm the coordinator of modules 1 and 4 in the master. So we're going to see the relevance of these 2 modules and understand what clinicians do on our day-to-day. Finally, I will highlight the importance of investigation and research on our results. Infertility is increasing each time more. In fact, the World Health Organization estimates that 15% of the couples will require assisted reproductive techniques to achieve pregnancy, will require our help at some point of their life. But it can be even more. The causes are multifactorial, but involved in this increase of infertility are some factors such as stress, contamination, and advanced maternal age. But it's important to study the couple as a whole, not only the woman, but the male and the female. We have to study both the male and the female, as commonly there is a multifactorial cause. So there are many therapeutic approaches, and we have to do a correct diagnosis so that we can offer the correct treatment. We have to individualize. Maybe we can start with a more simple treatment, such as intrauterine insemination, but it's not uncommon that we have to go to more complicated treatments, such as IVF with genetic diagnosis or even oocyte donation. And as I was saying, the dilemma of this century is that each time women have decided to delay the age at which they start with childbearing, mainly because they are looking for employment success. Also, there's an inadequate misconception that fertility treatments can reverse this infertility related to age, but this is incorrect. So, as I was saying, with age, there is on one side a decrease in the oocyte quantity, which would be a decrease in the ovarian reserve, but most importantly, and what we can't reverse, there's a decrease in the oocyte quality. And this is going to cause an increased aneuploidy rate, also an increased chromosomic alterations with an increased miscarriage rate. All this is going to cause a decreased live birth rate related to age. But in our clinics, we also see many other pathologies. Like, for example, we see 5% of implantation failures, recurrent miscarriage, and low ovarian response. Also, we see a lot of pathologies related with the uterus, as many women come to our clinics looking for a second opinion. So, as I was saying before, in the master, we're going to see there are two modules related with the clinical part. Module 1, we're going to see basic endocrine and physiologic of the reproductive system. We're going to see the reproductive system of both the male and the female. Instead, in Module 4, we're going to see these pathologies related to sterility. That, as I said before, are the pathologies that we see on our day-to-day. So our results have increased over the last years. This is mainly because our investigations have helped to increase these results. In IVI, we are not only gynecologists and embryologists, but we also have a large team of researchers that help increase these results. So this is why over the last years, we have seen an increase on our implantation rate and pregnancy rate. In fact, in IVI Valencia, there are over 20 clinical trials going on. With these clinical trials, we are not only looking to improve our results, but also we are looking to provide new techniques to help our patients. And this is reflected in the large number of publications that we have on all these journals. So in this master, you will have the opportunity to write a final master thesis. And if you want, and if you talk to your tutor and you're interested, you could even publish in one of these journals. These are all of the papers published over the years, consequence of our large number of investigations. So now I'm going to talk about ovarian rejuvenation. This is one of the researches going on in IVI Valencia, that its objective is to promote the ovarian rejuvenation in women with diminished ovarian reserve. The main objective is to help these women, in which the only potential treatment would be oocyte donation. So the objective is to restore the ovarian niche with the use of a stem cell derived from the bone marrow. So bone marrow derived stem cells could be an alternative to those patients with impaired ovarian reserve to restore their reproductive potential. In fact, in this woman, when a biopsy is carried out, we saw that there are many primordial follicles present. So the theory is that the correct ovarian niche is provided, these follicles could be rescued. And this is why we are using the bone marrow derived stem cells, because they could be easily, by administering GCSF, they could be easily got to the peripheric blood, and they could be easily obtained by a simple apheresis. So we carried out a first pilot study called ASCOT, Autologous Stem Cell Ovarian Transplantation. It was a pilot study with 20 patients defined as with low ovarian reserve as defined by the ESRO criteria. Only one of the ovary received the intervention, as the other ovary served as the control, and the follow-up lasted for 6 months. We measured the anti-Mullerian hormone and the antral follicular count. So results showed a positive response for both the antral follicular count and the anti-Mullerian hormone levels in 74% of the patients. There was no difference between the control ovary and the injected ovary. We saw more follicles developed and more oocytes obtained. Even though the euploidy rate was low, there were 3 pregnancies after IVF and 3 spontaneous pregnancies. In total, 6 pregnancies with 3 live births. But this population was quite heterogeneous because even though there was infertility, they had regular menses, low AMH, and low quality oocytes. So we wanted to move to a more homogeneous population that would be the premature ovarian insufficiency with menopause, no menses, FSH increased, and hypoestrogenism. So we carried out a second pilot study in 20 patients with premature ovarian insufficiency as defined by the ESHRE criteria. In this study, there were two arms of the study. The second arm was identical to the previous study, where they were given GCSF and the stem cells were in peripheric blood, and then they were recollected by apheresis and infused into one of the ovarian arteries. But the first arm, the cells were left on peripheral blood circulating. So preliminary results showed a positive response to ASCOT in 60% of the patients. There was no difference between both arms, mobilization and ASCOT. Interestingly, there was an increase in the antral follicular count more than 30 days post-treatment, which means that probably the primordial follicles were being recruited. The climacteric symptoms were ameliorated in 50% of women, and 60% of women had their menses back at some point of the follow-up. There were more oocytes retrieved, 2 embryos, and 1 pregnancy after IVF in the ASCOT group with 1 live birth. So there were many clinical trials going on with platelet-rich plasma infusion, as you can see here. So our group carried out a pilot study in animal, combining the platelet-rich plasma with bone marrow-derived stem cells in induced premature ovarian insufficiency. We saw how there was an accelerated restoration of ovarian function compared with the other techniques by themselves, with the bone marrow-derived stem cells by itself and the platelet-rich plasma by itself, with more neovascularization, a reduction of apoptosis, and increased folliculogenesis. So now we developed a third pilot study in humans that is now going on in IVI Valencia and in IVI Madrid, where we are combining both techniques, the ASCOT and the platelet-enriched plasma. We hypothesized that the sequential combination of the non-cellular compounds of ASCOT and the plasma could induce a positive ovarian response. So this clinical trial is now going on. We have to recruit 42 women, and to date we have recruited 35 women, and we are waiting for results. So to conclude, these are a series of investigations using both the bone marrow-derived stem cells and plasma with promising results. And even though we still need more investigations and a longer follow-up, the idea would be to validate this technique for a real therapeutic alternative in patients with diminished ovarian reserve, where their only therapeutic option would be oocyte donation. In IVI, as I said before, our investigation favor the progress on our day-to-day. So thanks to all our investigations, we can offer our patients better results and novel techniques, all based on scientific evidence. So thank you very much. And now,
okay, thank you very much to both of you, to Dr. Marcos Meseguer and Dr. Nuria Pellicer. I have been receiving some questions that I have been answering online, because many of them were just an issue to redirect to the right email address or the right website in order to look for the information requested from the audience. Let me check I think all the 4 questions you submitted have been personally answered. We are just waiting for about 20-30 seconds more, just in case anyone has any other question to submit. Otherwise, we will close this session just thanking the professors for their participation, the technical support from IVIRMA Education, and all of you for attending this session today with us. And I hope to see you all in our next Masters edition. Just in case you might need some additional information, I'm sharing with you and I'm leaving the slide showing the way to connect with the Masters management from the teaching department with elena.garay@ivrma.com or my own email in order to respond any other question you may have. Thank you, everyone, and I wish you luck and stay well. Thank you.